Friends, welcome to Biology Made Easy. We are discussing the different aspects of a mutation. In one of the videos, we have discussed what are the different mutagens, then the classification, and then aneuploidy. Then in another video also we have discussed what are the different examples of euploidy and chromosomal aberrations. But today in this video, we will discuss particularly the gene mutation. Gene mutation we will discuss. So, gene mutation is a sudden change in the base sequence of the DNA that changes the characters of the organism. What is the basic difference between the genomatic and chromosomal aberration and gene mutation? Usually the chromosomal aberrations and genomatic mutation, that is polyploidy, they are observable from outside. But the gene mutation is not observable because it changes only a single base pair, but its effect is far-reaching, but we cannot see it. When we can see it, only when it will be in a recessive condition and it will express itself. Usually, most of the mutations are recessive and they can express themselves. Only when they will come to homozygous condition, then we can know that actually some mutation has taken place. Those mutations which can be immediately observed, they are dominant. But the dominant mutations are very rare and they are lethal. So most of the mutations are usually recessive and not observable. Only in future generations, when only homozygous condition will come, we can know that some mutation has occurred. Now this gene mutation is again divided into substitution mutation and frameshift mutation. The gene mutation, we can discuss it in two headings. One is the substitution mutation and the other one is frameshift mutation. Now the substitution mutation, again it is of two types. It may be transition or it may be transversion. It may be transversion. So, and this frameshift mutation, this can be deletion and insertion. So, let us discuss one by one. Let us first discuss this number one. This is the transition, and it is a type of substitution mutation. In a transition, what happens? We know that there are 4 nitrogen bases: purines and pyrimidiన్స్, adenine, guanine are purines, thymine, cytosine are pyrimidiన్స్. So when we will call it a transition mutation? When one purine will be replaced by a purine, or a pyrimidine is replaced by a pyrimidine. That means A and G, they can alternate with, that means A can be replaced by guanine or guanine can be replaced by A. Similarly, thymine can be exchanged with cytosine or cytosine can be exchanged with thymine. That means a purine is replaced by another purine or a pyrimidine by a pyrimidine. That is known as a transition mutation. But the second type is transverse mutation. In case of transverse mutation, a purine is replaced by a pyrimidine and vice versa. Pyrimidine is replaced by pyrimidine and vice versa. So, how it is taking place, you see. Suppose this is adenine and this is guanine. This is thymine, this is cytosine. That means how the change is taking place? That means A can be replaced by T, T can be replaced by A, because this is purine, that is pyrimidine. Similarly, A can be replaced by cytosine, cytosine can be replaced by adenine. Similarly, now the guanine can be replaced by cytosine, and guanine can be replaced by thiamine, because so this is a purine and these two are pyrimidines. So when exchange takes place like this, a purine is replaced by a pyrimidine or vice versa, this is known as transverse mutation. So in case of transverse mutation, usually one common example will take the example of sickle cell anemia. Sickle cell anemia is a good example of this transverse mutation, because in case of sickle cell anemia, what happens? This is the normal hemoglobin and this is the sickle cell hemoglobin. Now usually the DNA in case of normal hemoglobin, it is GAG and CTC, and in this case it is GTG and CAC. The only change is this T is replaced by A. That means T means thiamine is, and this is pyrimidine, this is purine, that means pyrimidine is replaced by a purine, so that this is transverse mutation. Now, next what happens? Now in case of RNA, now the RNA will be, this one RNA will be GAG, and here it will be GUG. Now, this, according to genetic code, this GAG, it will give rise to glutamic acid. Glutamic acid, while this will give rise to valine. So usually, this is present in the 6th position of one of the beta chains, while this will be amino acid number 5, then amino acid number 4, and likewise amino acid number 1. This also in the same case, this the 6th position, and again we will get amino acid 1, 2, 3 in this side. That means when glutamic acid is present in the 6th position in one of the beta chains, we get normal hemoglobin. But when it changes to valine, and why it changes to valine? Because in the DNA, thiamine is replaced by adenine. So you see, the sickle cell anemia is a case of substitution mutation. It is a case of a transverse mutation. It is a case of also gene mutation. So this is one of the examples of this transverse mutation. So this is about transition transversion. Now let us discuss deletion and inversion. That is frameshift mutation. Now in case of frameshift mutation, there is no substitution of any of the bases. Here either due to deletion or insertion of nucleotides, the entire frame of the DNA is changed. That means, see, when the frame is changed, there is far-reaching effect. That is why the frameshift mutation is more dangerous than even this substitution mutation. And the frameshift mutation, it usually responsible for the thalassemia disease, thalassemia and muscular dystrophy. So these are all serious diseases caused due to frameshift mutation. That is why I already have told you that frameshift mutation is more dangerous than that of the substitution mutation. So how the whole thing is happening, let us see. Now let us discuss the deletion. Now in case of deletion, what will happen? Suppose let us take any example of a segment of a DNA. Suppose one segment of DNA has T, A, C, G, A, G, C, T, A, G, G, A, C. Suppose this is the normal DNA. Now suppose there will be deletion. So deletion means anywhere. Suppose you can take that this is deleted. Suppose this is deleted. Then what will happen? Now it will be, after deletion, it will be TSC. Now this G is deleted. So it will be AG. Now this C will come to this side. Then it will be TA, and this G will come to this side. Then AC. Now you see, from the point where it is deleted, the remaining codons are changed. So when there will be deletion of 1 or 2 nucleotides, it will have far-reaching effect. But if the change will be in multiple of 3, then it will have less effect. Because multiple of 3, if you completely, the codon will be deleted. Because 3 bases make 1 codon. So if that will be changed, that will have, because 1 amino acid will be deleted. But in this case, all the amino acid will be changed, and the enzyme, the protein which will form, may be completely non-functional. So this is known as deletion. If you take the case of insertion, in insertion again also you see, let us take this same example, this TSC, GAG, let us take, and somewhere will be some added. So this is the example of deletion. Now let us take the case of insertion. So in insertion again, suppose TSC, and now we insert some. Suppose here T is inserted. T is inserted here. So now what will happen? When T is inserted here, then it will be TGA. Here T is inserted, so TGA. Then G will go to that side, GCT. Then A will move, AGA, then C. So you see, whether it is deletion or insertion, in both cases these sequences are changed. But what is the far-reaching effect? You cannot guess unless I give one analogy. So let us give one analogy. Let us give one sentence where all the words have 3 letters like that of the codons. Suppose the cat ate the rat. Now this is a meaningful sentence. The cat ate the rat. If there will be deletion or insertion, what is happening? Suppose here this C will be deleted. So when it will be deleted, what will happen? Now it will be THE, then C is deleted. That means AT, A will come to this side. Then TE, this T will come to this side. Then HE, the R will come to this side. Then AT. So you see, previously the cat ate the rat. It is a meaningful sentence. But when there is deletion of one letter, now it is the, AT, TET, HER, AT. That means there is some only jumbling of letters. It is meaningless. Similarly, if there will be addition, so if there will be addition, anywhere you can add anything. Suppose THE, now you add something here. Suppose let us add G here. We'll add G here. Insertion. So what will happen? Now it will be G, C, A, then T will move, T, A, T, then E will move, E, T, H, then E, R, A, then T. Again it is meaningless. So that is why I have told you that deletion and insertion have far-reaching effects and they are more dangerous. You can guess what a drastic change is taking place when there is a change in the frame of this DNA. So this is a deletion, this is insertion. In one DNA, there can be both deletion and insertion simultaneously. Suppose this is the cat ate the rat. At one place there may be deletion, at another place there may be addition. So again, that is the same type of change will take place. So that is, this has far-reaching effect. As I have told you that the thalassemia and muscular dystrophy are examples of frameshift mutation. So we have discussed this substitution mutation, frameshift mutation. Then there are some other examples like what is a nonsense mutation, what is a missense mutation, and what is the Samesense mutation, which are also very important. And we'll explain with examples. So mutation can be nonsense mutation, it can be Samesense mutation, or it may be Missense mutation. Nonsense mutation means when due to change in the one of the bases, it will completely stop. That means there will be no protein synthesis. Samesense mutation, that means mutation will take place, but the result will be same. That means it has no effect. Missense mutation, due to change in one of the bases, the amino acid changes. So it has far-reaching effect. So in this case, the amino acid changes. Now let us take one example so that our understanding will be better. Suppose let us take one example of UGC. UGC, don't think that it is a University Grants Commission. It is one of the codons of the genetic code, UGC. And this UGC usually it codes for cysteine amino acid according to genetic code. Now suppose this is C will change. Now this is C will change, and it will change to what? It will change to U. It will change to U. So the remaining two will be UG. Now it will be UGU. And both UGC and UGU, they are synonyms for the same amino acid, that is cysteine. Now you see, there is change in the nucleotide. That means mutation has taken place, but the amino acid didn't change. That means even after a mutation, the effect is same. So this is known as Samesense mutation. This is known as Samesense mutation because there is no change. Now, suppose this C will change to A. Now it will change to A. So what is happening? Now it will be UGA. And according to genetic code, UGA is stop signal. That means when C will change to A, that means the protein synthesis will be stopped. So it is known as a terminating codon or nonsense codon. So this is known as nonsense mutation. So this is the nonsense mutation, this one. Now suppose this will change to, the C will change to G. It will change to G. So when it will change to G, what is happening? Now this UG will remain. Now this UGG. And when it is UGG, according to genetic code, this is responsible for tryptophan. So see, previously it was UGC, it was representing cysteine, but due to mutation in this sequence, now it becomes UGG, which is responsible for tryptophan. Now, instead of cysteine, it will bring tryptophan. That means this is known as missense mutation when the amino acid changes. So this is missense mutation. That means from one example, UGC, we can take how the nonsense mutation, the Samesense mutation, and missense mutation is taking place. This is the easiest example to remember for examination point of view. So these are the different types of the gene mutations, the substitution mutation, the frameshift mutation, and this nonsense, missense, and Samesense mutation. Then let us discuss two questions before closing the discussion. So one question is MCQ. One point mutation involves A choice deletion, B choice insertion, C choice change in a single nucleotide, and D choice duplication. Gene mutation involves. Now the second question. Second question is sickle cell anemia is an example of one is substitution mutation, B choice transverse mutation, third choice is Missense mutation, and the fourth choice all of the above. So we have discussed all these things. I think you will be able to answer both the questions. So thank you very much for watching the video. God bless you and have a good day.